Megan said, today I'm going to talk to you a little bit about my work that I've done with milk nanovesicles. I don't know if you attended a few months back, Martin, another EV scientist, presented his data on this new field of research that we're all embarking in on extracellular vesicles that's found in mammalian milk. So I'm going to tell you a little bit about that today. So I don't have to tell you all about the magical components of milk that are both nutritive and non-nutritive. It would be preaching to the choir here for me to go through this slide, but of course, milk contains a plethora of components that are both nutritive and non-nutritive, and these are influenced by different variables in terms of maternal factors or environmental exposures or the different exposures during early life. And I'm looking specifically at one type of those bioactive components that are called milk-derived EVs or milk-derived extracellular vesicles. So what they are is that they are small lipid droplets. So they're fat droplets that are 30 to 150 nanometers in size. They contain a very stable lipid bilayer. So something that resembles that of our plasma membranes, our cell membranes, our mitochondrial membranes. The organelles that are within our cells contain very similar structures to these EV membranes. And because of this stable bilayer structure, it provides EVs the ability to survive intestinal degradation. So gestation, like digestive tract breakdown, as well as salivary breakdown. And they have been shown to survive the consumption in neonates, especially in early life. And they have also been shown to cross intestinal barriers. So they get through the intestinal endothelium into the central circulation, and they are being uptaken by the peripheral organs and tissues. So they've been found in the heart, in the liver, the kidneys, the spleen, even the bone marrow of neonates in early life. And they've also been located in the brain. So they have been shown to cross the blood-brain barrier and get to different parts of the brain. And the really important things about these EVs is that they carry cargo. So they carry information in the forms of lipids. So they could be short-chain fatty acids that could provide pro-survival effects and anti-inflammatory effects. They have been shown to contain peptides, small hormones that can potentially regulate growth and development. And they also contain a group of RNA that are called microRNA. So microRNAs are very small 18 to 25 base pair RNA molecules that are non-protein coding, meaning that they don't code for protein production, but they participate in protein translation. So these guys, they regulate what type of mRNA transcript will ultimately become functional protein. So they're sort of a transcriptional, post-transcriptional regulator system that's found in the cell. And up to date, 1,500 different kinds of human-specific microRNAs have been found in these milk EVs. So, and these milk microRNAs, they could be conveying biological effects. They could be inducing developmental programming in offspring once it's been released into the cytoplasm of the cell. So how do I isolate these milk EVs? There's different methods that you can employ to isolate milk EVs. The one that we use in my lab and that I've used for the past several years is the differential ultracentrifugation method, meaning that we use different speeds of centrifugation. We spin these samples at different speeds for different time periods over a very long period of time. Sequentially increasing the speed and the number of revolutions, and we can isolate the different components of milk this way. And I typically work with unpasteurized mammalian milk. I do not do the pasteurized version of it because some work has been shown that pasteurization could impact EV survivability, depending on the type of pasteurization, whether it be like whole pasteurization or full-on the long-term pasteurization. So we spin these whole unpasteurized frozen milk for different periods, for different speeds, and then we isolate different components along the way. So for example, cream and fat cells, milk fat globular membranes, milk proteins, maternal cells that are in the milk, these get periodically removed. And we also precipitate out casein. Casein is a specific kind of protein. It's a major component protein in milk. We also precipitate out casein, and we only look at the whey portion of milk, and we isolate the EDs from the whey portion. So once we do that isolation, we should have a pure to semi-pure isolation of EDs. And then, of course, we have to characterize those EDs to make sure that we're actually looking at extracellular vesicles, not apoptotic bodies, and not broken pieces of particles or cell debris. So we use different kinds of methods. One of them is transmission electron microscopy. So these are images that are taken from different sources of milk. These are EDs that were isolated from, for example, bovine milk. We have the human milk, and then we have rodent on the bottom here. As you can see, how these milk EDs show up in an electron micrograph is that you see that outside lipid bilayer stained, and then you have the inside cargo that's stained. So they kind of look like sunny side up, cute little eggs. That's how they show up. And we also get glimpses of broken EDs in our samples as well. So if you look at the rodent TEM image here, you can see some of these smaller little vesicles also in pieces. That tends to happen with manual isolation methods. Then after we do transmission electron microscopy, we use another method called nanoparticle tracking analysis. So essentially what this does is that it zones in a laser onto a stream of EDs that's suspended in water or PBS, some sort of a saline solution. And depending on the reflection and refraction coefficients of your EDs, it can determine how much EDs you have in your system in that solution, and also the particle distribution. So how big or small those EDs are, because we can have EDs going anywhere from 20 to even upwards of 500 nanometers based on the type of EDs we're talking about. So each of these dots here, if you can see that's lighting up, those are milk EDs that was from a human isolation. Oh, sorry. I think I have to manually go to the next one. And then we also look at the abundance of surface markers on these ED fractions to make sure that, again, we are looking at actually EDs and not apoptotic bodies. So we look at the abundance of a group of proteins called the tetraspanin proteins. So these are like the CD63, CD9 proteins that are abundant in the ED bilayer. And what they're showing you here is a Western blot image, immunoblot image of human EDs. And you can see that in the CD63 images, the EDs are lighting up. So we see that they're abundantly expressed. But however, in this calnexin profile, calnexin is a Golgi apparatus protein. So it should not be showing up in EDs because EDs should not have organelles. So this is showing us that there's very little to no cellular contamination of our extracellular vesicles. That is a pure fraction that can be used for downstream investigation. So given the novelty of the field, there's so much that we don't know about EDs, even though the knowledge gaps are being filled every single day. More and more people are starting to work on milk EDs, but there's still quite a bit that needs to be explored, which is great for me as an early career researcher. This is what I intend to do for the next several years. So my long-term goal with EDs is to really understand their functional effects in offspring once they're being uptaken and the cargo is being released. So do they convey pro-survival benefits to offspring? For example, can they be thought of as sort of a rescue effect or remediation a route that's happening in postnatal life to mitigate some of the exposures during in utero life, right? To maternal stresses. And another question is that is milk ED composition sensitive to maternal factors? So things like immune activation or maternal diet or psychosocial stress, do they also change the composition of these EDs in milk, similar to some of the other components that are sensitive to maternal factors? Another question is that do brain cells uptake EDs? Yes, they go across the blood-brain barrier, but do they get actually uptaken by neurons and astrocytes and microglia, different components of the brain, right? And if they are being uptaken, what are some of the developmental outcomes of its transfer? What are the functionalities of the cargo that they carry, right? And what are some of the acute versus prolonged changes that's been incurred to the host through this transfer? So for today's talk, I'm gonna go through some of the work I did with trying to assess whether MED composition is sensitive to maternal factors. And I'll also show you a little bit of data on whether brain cells uptake and localize EDs, and do they do anything in the brain. So to answer the first question of whether milk EVs are sensitive to maternal factors, I used a diet manipulation model. So I wanted to use maternal diet as that early life stressor to shape the lactation dynamics, especially the milk EV lactation dynamics. So we used a rodent model, long Evans rodents, and we put them on the females on a high fat diet. So this diet has 60% saturated fat. And they were on this diet for 4 weeks prior to mating. And then they remained on the diet through gestation as well as lactation period. So they were on the diet for a total of 12 weeks. And then we collected the milk from the dams at 7 days postnatally, as well as we collected tissues from the pups. So we collected the brains, we collected the peripheral tissues to check what type of regulation, cellular regulation, is being seen in the pups as well as the mom. So we had two groups of experimental conditions here. We had the high fat diet group, and then we have the control group, which contained both the pups and the mom. So we used female and male offspring in our cohort. And we collected all these different kinds of tissues, as well as stomach milk, serum, visceral fat from the babies. So and my colleague, Dr. Abuesh, or Sam Abuesh, she also reported that with her studies, moms who are on this high-fat diet, the rodent moms, they tend to spend more time nursing their pups compared to the control counterparts. So what this means is that during the dark phase, which is their active phase of the life cycle, they tend to spend more time nursing their pups, and they tend to be on the nest much more at a higher percentage than the control counterparts. But she did not notice any differences in terms of other maternal care behaviors between moms that were on a high-fat diet versus the control metabolic group. So I wanted to see that, okay, so the high-fat diet moms are nursing their pups more often and for longer periods. Does this also convey to changes in the milk EVs that's been received by the pups, right? Or that's present in the milk itself? So when we looked at the milk EV profiles, or the amount of milk EVs present in high-fat diet milk versus control milk, what we saw was that there's actually an increase in the concentration of milk EVs in the high-fat diet milk versus that of the control milk. And these are also the TEM images of the milk EVs that were isolated from the high-fat diet on the top, and then the control diet on the bottom. And this is how we collect milk from the rodents. Yes, I collect milk from really small rats, and it's great. So now that we found out that the milk EVs are also is higher in the high-fat diet milk, as well as the moms are spending more time nursing, we wanted to check the cargo. So is the cargo changing in the milk, and how is that cargo causing any kind of developmental effects, if any, right? So to do this, we looked at a specific family of microRNAs that are highly abundant in milk. This microRNA 148, 152 family is considered one of the top microRNAs that are abundant in all types of mammalian milk, not just human, but in rodents, in bovine, even marsupials have this family of microRNA as one of the top 10. And this family of microRNAs, they're also really great to use as a tool to see these kinds of effects, because they do one-to-one regulation of functional effects. What I mean by that is that if you can measure the microRNA abundance, and then you can also measure their direct targets to see whether there's any kind of downstream effects taking place from the differential levels of microRNAs present in the milk. So the type of mRNA, or their direct targets we looked at for this study, were the DNA methyltransferases. So DNA methyltransferases are enzymes that regulate methylation levels or DNA methylation levels in the body. And DNA methylation is a mechanism that regulates gene expression. So whether genes are turned on and off is determined based on the amount of methylation that's present in the promoter region, amongst other systems, but mostly promoter methylation. So we looked at this microRNA 148, 152 family along with their direct targets, the DNMTs, in these pups, as well as the milk, to see what's happening to the cargo. So what we first found was that when we looked at the stomach milk of the babies, so both female and male offspring, we see that microRNA 148 3P levels were significantly lower in the stomach milk EDs of both females and males. That were born to high-fat diet mothers and that were exposed to high-fat diet during in utero compared to their control counterparts. Then we also followed those microRNAs into the brain, and we looked at different brain regions like the hippocampus, the prefrontal cortex, as well as the amygdala. And we saw that in the amygdala, the females actually had the lower levels of that exact specific microRNA. However, the males did not show any differences. So we see that this microRNA, this milk microRNA, is potentially being regulated in a maternal factor specific manner in the offspring. So then we looked at the targets that those milk microRNAs are responsible for regulating, the DNA methyltransferases. What we saw was that the DNA methyltransferase 1 levels were actually increasing in the amygdala of those same female offspring. So we see that inverse regulation that we expect to see with milk microRNAs and with their targets. If the milk microRNA levels are lower, then typically the targets that they regulate, their levels should be higher. So they should be more abundantly expressed or available in those tissues. We also saw that methyl CpG binding protein, which is a protein that DNA methyltransferase 1 works with in unison to carry out methylation, was also upregulated in female brains or female amygdala in response to the in utero high-fat diet exposure. Then we also went one step further and we looked at the protein levels and enzymatic activity, because these DNA methyltransferases, they are enzymes. They are catalytic in the cell. So when we measured their enzymatic activity, we again see very much a similar correlation here, where we saw increases in enzymatic activity of DNA methyltransferases. And then when we looked at the global methylation increase in the amygdala of the female offspring, we also see an increase in methylation. And there's also a very strong correlation between global methylation levels in the brain versus that of the DNMT enzymatic activity. So it seems like in this female offspring, the milk EDs are causing epigenomic effects in the tissue. So what does this mean in terms of development, right? And why should we care that these epigenetic signatures are changing? So to figure out which developmental pathways these DNA methyltransferases and these methylation patterns are regulating, we did what's called reduced representation bisulfite sequencing. So this is a sequencing method that you can use to look at regulatory regions. So I'm not looking at the entire genome to look at entire methylation patterns, because this method allows me to look at much more of the promoter-specific methylation. So we can pinpoint which genes are being upregulated, which genes are being downregulated, and what does that mean in terms of the pathways that they control. So when we looked at the number of promoters that were differentially methylated in the high-fat diet exposed offspring versus that of the control diet exposed offspring, we saw that 720 promoters were differentially methylated, and they were hypomethylated. So what this means is that we have increases in regulatory mechanisms that are associated with neuronal projection and nervous system development, for instance. Metabolic regulation was one of those also pathways that were differentially methylated in these babies. And cellular morphogenesis and development, apoptosis, so cell death, and gene expression and regulation were also some of those other pathways that were regulated through methylation in the female amygdala. these patterns were not present in the male amygdala. So females were uniquely sensitive to this maternal diet exposure compared to their male counterparts, their littermates, essentially their siblings.
Sanoji, I just want to mention that we have 5 minutes left, and I want to make sure there's time for questions.
Oh, absolutely. Let me just quickly show you a few other slides, and then I'll stop questions. So that's a study that we looked at whether maternal factors can influence milk composition or milk EV composition. Then we also wanted to take that one step further and see, okay, well, we see these transcriptional differences and protein differences in the brain of these pups with these milk EVs, but can we mechanistically show in a cell culture model how these milk EVs are being uptaken? So to do this, we use a microglia, which is the workhorses of the brain. I call them the MVPs of the brain, not the neurons, the microglia. So we grew them in a cell culture, and we introduced milk EVs to them with fluorescent tests. So I'm just going to go through here really. Oh, I'm sorry. This should be animated. Oh, my fault. Okay, anyway, sorry. So what we did was that we isolated the milk EVs and we tagged them with fluorescent tags, and we introduced these milk EVs to the microglia cell line, and we imaged over a live imaging period of 24 hours how these milk EVs are being uptaken through endocytosis into the cytoplasm of these cells. And what we saw is that we first isolated and tagged the RNA composition of these milk EVs. And if you look at this image here, what you see is that these microglia are absolutely uptaking these milk EVs, and they're being localized actually in different parts, including the nucleus. So they're in the cytoplasm and they're going into the nucleus of these microglia cells. So they're getting in there, getting into the brain cells. And we also repeated that experiment with protein tagging. So we tagged the proteins that are inside these milk EVs, did the same experiment with the same microglia cell line, and we got the same results. So we got the same type of localization pattern of these milk EVs into the microglia. So the second part of the experiment shows you that milk EVs are indeed being uptaken into brain cells, and they are potentially participating in downstream function. So I will stop here because it's 1:58, and I'm happy to take more questions.